Thank you very much, and thank you to the Foundation Jung for this award and for how it worked, and for inviting me to this, giving this talk and this seminar. You have heard to the magnificent work of Dr. Roof, and what I will do is go a few years earlier and travel, I hope, with you in a journey from what made that possible and from where did the tumor immunology come. And although immuno-oncology and immunotherapy is a long story, the modern tumor immunology, it became respectable a few years ago. And I will discuss how, from immune response to cure, immunotherapies are transforming the life of cancer patients. 2000, so 25 years ago, when Annan and Weinberg described the hallmarks of cancer, there was not a word on immunology. So you need to be insensitive to anti-growth signal, to evade apoptosis, to proliferate, etc. But nobody cared about the immune system. 10 years later, 2011, the same authors had 4 emerging hallmarks, 2 of them being associated to the immune system: avoiding immune dysfunction and tumor-promoting inflammation. So what happened in these 10 years? So there's been a lot of basic science, and I would like to emphasize the fact that if Dr. Roof is capable now to treat patients, it is because basic science has transformed that field. And among this, I will illustrate what has been done in my own laboratory. What we have been studying is the tumor microenvironment. And we have been analyzing the organization of the tumor microenvironment with immune cells in the tumor core, T cells, B cells, macrophages, whatever, in the invasive margin, and we describe these tertiary lymphoid structures that I will come back to it, which we believe are the sites in which anti-tumor immune reactions are being generated. The seminal work in that was done 20 years ago in colorectal cancer. Jérôme Gallon was the first author. We analyzed a series of 1,000 colorectal cancers, and this is the disease-free survival depending on what the pathologist classifies the cancers in the Duke stage. Yellow, stage 1, is small non-invasive cancers. Green is local invasion. Red is lymph node invasion and metastasis. And you see that you have a gradual 20% relapse with small tumors, 100% with metastasis, and 40% when they have lymph node invasion. We analyzed all the cells of the immune system in an unbiased approach in the tumor microenvironment and in the core of the tumor and outside. And what we found is that there was a category of cells called the T cells that have been discussed, which recognize peptides presented by MHC class 1, and memory T cells in particular. When patients had a high density of these cells, even patients with lymph node involvement did better than patients with low, with small tumors. But more importantly, when these T cells were lacking, even patients with very small tumors behaved as metastatic patients and relapsed very rapidly. And at that time, we dared to write that the evaluation of the type, the density, and the location of the natural, this is before any treatment, in site immune reaction with a distinct tumoral territory predicts clinical outcome better than the TNM classification, better than the tumor-associated markers. I can tell you we were not popular at all at that time, and it has been a long way to make this field a respectable field. However, Very rapidly after our work, there has been a lot of other works showing that indeed cytotoxic T cells, CD8 T cells, particularly when they are memory, are of good prognosis in red, in green here, in the vast majority of cancer, where the density is high. Few cancers is not the case, I have no time to discuss it, but in general, it is of good prognosis. So if an immune density is important to predict clinical outcome, where is the immune reaction shaped? And again, we were lucky to make a discovery, which was known from in the fields of inflammatory diseases or graft rejection, which is the presence, this is in lung cancer, around the tumors of lymphoid aggregates that we call tertiary lymphoid structures. And you see here in lung cancer, you have a lot of these surrounding the tumor beds. And when we analyzed what is their impact, in green are the T cells, we found that the presence of these TLS, tertiary lymphoid structure, were the most cancers also of good prognosis. In HCC, it depends. If they are in the nodules, they are of good prognosis. If they are distant of the nodule, they are of bad prognosis because they are in the inflammatory cirrhotic liver. Macrophages usually are not of good prognosis. Tregs, it depends on the inflammation. So if TLS are of good prognosis, do they shape the immune reaction? Can we show that when they are present, the immune infiltrate is different? And we looked here at, we developed a software called MCP Conta, and by transcriptomic we can measure in the various cells within a tumor, and this is spatial transcriptomics. So this is a tumor, and we can analyze the gene expression at each spot of this tumor slide, of the slide with the tumor, and so know where is the cells that corresponds to this immune signature located. So in a tumor, when there is no TLS, you see this is kidney cancer, we see the tumor cells by the expression of CA9 as a tumor marker. There is no B cells. B cells are the cells that you will see differentiate into the antibody-producing plasma cells. There are T cells. There are CD8 T cells, the cytotoxic ones. There are NK cells. We didn't look at MATE. There are myeloid cells. There are fibroblast endothelial cells. But it's an unorganized tumor microenvironment. It is dispersed. When you have TLS, you see them here in the H&E slide, you have B cells now. And they are mostly in the TLS, although they are also outside. T cells are also enriched in TLS and outside. The other cells are scattered into the tumor, with the exception of fibroblasts, which are also surround TLS and are found outside. But now you see the tumor microenvironment is an organized one. It's not just dispersed, it's really the organization governed by the TLS. So if B cells are hallmark of TLS, what type of B cells do we have? Very few naive B cells, mostly memory B cells who have encountered the antigen already, germinal center B cells, plasmablasts, the cells just before the plasma cells that produce antibodies, and plasma cells, which we find in the TLS and outside. Since plasma cells produce antibodies, can we show that there is antibodies, immunoglobulins in the tumors? And the answer is yes. Pan-immunoglobulin gene expression is increased in TLS and outside. IgM, the unswitched immunoglobulin, the natural reaction is located into TLS, but IgG and IgA, the post-switch mutated immunoglobulins, are in TLS and outside of TLS. Indeed, by immunoimaging, we showed that here's the TLS. If we label with anti-MUM1, which labels plasma cells, and IgG or IgA, producing IgG and IgA, we found that leaving the TLS, there are plasma cells, and they leave it embedded in fibroblastic conducts. At distance of TLS, you see here a fibroblastic conduct that seems empty, but if you label it, it's full of plasma cells producing IgG and IgA, and they are attracted there by a chemokine called CXCL12. Does it have an impact? Jens says yes. When you have TLS in green, you have many high density of IgG-producing plasma cells, also IgA, but less. But this is the important thing, that people, it was the elephant in the room. Nobody was looking at it. But if you look at the tumor, you see in green is labeling the tumor, in red is anti-IgG antibodies. These tumor cells are covered by IgG antibodies. And the same cells have both markers. When you have no TLS, you have no IgG. And when you have IgG on the tumor cells die. You have apoptosis measured here by cleaved caspase 3, and this is due to macrophages in this case. In white is dying tumor cell caspase 3, red is IgG, and around we found macrophages killing or phagocytosing these tumor cells. We didn't find NK cells in this model, but it may exist in others. So let me come to the second part, and it's been alluded to already. 2011 changed the world because coming from two very basic experiments, and this is a Nobel Prize given to two friends of me, Jung-Stiftung Tatsuko Onjo, who discovered the role of CTLA-4 immune checkpoint inhibitors in 1994 and PD-1 in 1992, both in very basic analysis, trying to understand how T cells are activated and how T cells are selected in the tumor, and the thymus for PD-1. What are CTLA-4 and PD-1? They are expressed on activated tumor cells. They bind ligands, CTLA-4 the same as the activating receptor CD28, PD-1 another receptor, ligand PD-L1 or PD-L2, expressed on tumor cells or antigen-presenting cells. And this induces inhibitory signals through a motif that my laboratory had described, I-team sequence 30 years ago. So what Jung-Stiftung came about, and also Tatsuko Onjo, is that if you have antibodies to these interactions, you will block the inhibitory signal and reinvigorate the immune reaction. And indeed, it changed the paradigm of cancer treatment. Up to 2011, everybody tried to kill the tumor cells. And from then, people started really to activate the immune system, not caring of the tumor cells, and it may be pan-tumor approach, and I will discuss it. So we have 10 years. I told you that 2011, so now we have 10 years in which melanoma patients, advanced melanoma patients were treated with anti-PD-1 and CTLA-4. It has been published by Chad Wolfshock in New York. When I, as a young medicine clinician, treated melanoma before that, the metastatic melanoma, the expectancy of life of these patients was less than 1 year, 6 to 8 months. Now, you can see that this is the overall survival and disease-free survival. Treated with anti-CTLA-4 alone, 20% after 10 years. Treated with ipilimumab, anti-PD-1 alone, 47%, and together 43%. Half of the patients are alive at 10 years. But more importantly, if you take the patients that are alive at 3 years and you follow them for the other 7 years, they are cured. There is no relapse in these patients, or very few, you see. 100%, 88%. So one can say that immunotherapy has changed their life because they cured patients. And the question is, should we continue to treat them? Should we stop it? But these are good questions as compared to the questions that we had 10 years ago. Now, it was said not everybody responds to immunotherapy. It will come. You will re-invite me in a few years from now. And maybe I will have another speech, but today it's not the case. So what are the markers that the most strongly are associated with response? PD-L1 was an obvious marker, but it's not a good one. One good marker, since it reinvigorates T cells, is that the presence of CD8 T cells in the invasive margin in this case, and in the tumor core before and after treatment. And this is work by Tony Ribas in melanoma, showing that in responding patients, you had CD8 T cells, and they enter into the tumor from the invasive margin. In non-responding tumors, you don't have that. What do T cells recognize? They recognize mutated peptides. And there are a series of cancers in which there are deficiencies in the genes that code for DNA repair enzyme. And we call them microsatellite unstable cancers. Most are colorectal, but there are also cancers as well. And these MSI cancers, you see, this is a shrinkage of the tumor. Whatever the histology is, respond very well, in this case to anti-PD-1, with complete responses already here. And this led to the first approval by the FDA of a biomarker independent of cancer histology. If you are MSI positive, you should be treated by immunotherapy, whatever your cancer type is. And there is a strong correlation between the objective response rate to immunotherapy and the mutations. So what should we do with the people in the tumors that do not have a strong mutational rate? And then the other big part of the adaptive immune reaction came out. And a few years ago, our group in sarcoma, in collaboration with MD Anderson in melanoma, another group in melanoma, again, we analyzed again with the same approach various immune cells, and we found that the B cells and the tertiary lymphoid structures are strongly associated with immunotherapy, and we extended that in a pan-cancer analysis. And I show you only one slide in it. If you have here in the discovery cohort, here there were 540 tumors, 18 different tumor histologies, and we defined what we call mature TLS with the presence of a germinal center, the ones that are capable of generating plasma cells by the presence of follicular dendritic cells. And you see when there were no TLSs, the response rate in green was 20%, immature TLS the same, and it was doubled when there were TLSs. Again, longer progression-free and overall survival, and this was confirmed in two independent cohorts with a tripling of the response rates. Based on that, we did the first prospective trial in soft tissue sarcoma, a very aggressive metastatic disease, non-responsive, or it was considered non-responsive to immunotherapy. All patients in this trial have TLS. And Antoine Italiano in Bordeaux did that trial, and you see the response here. In an unselected trial, in the same institution, same patients, response rate was 2.4%, and non-progression rate at 6 months was less than 5%. In the selected population, response rate was 30%, 33% stable diseases, and 49% non-progression rate at 6 months. And you see once a stabilization or response is induced, it is lasting. So again, you have changed the outcome of the patient. Now all patients have TLS. What is the difference in terms of gene expression between TLS responding to tumors, to immunotherapy on the right part versus non-responding? Again, the immunoglobulin gene. Remember, sarcoma are low mutational birth rate tumors, and plasma cells are enriched in responding patients. So we look with anti-IgG. We could label plasma cells with IgG, but also, again, we label the tumor cells. So these plasma cells do produce IgG, which bind to the tumor cells. And what happens when you have IgG bound to the tumor cells? In renal cell cancer, when we separated the tumors with low IgG bound to them too high. The response rate was 62% when they have high binding versus 18%, longer progression-free survival, and they combo PD-1 CTLA-4, or even if we had the PD-1 treated patients alone. And in fact, it opened a field in which suddenly many people discovered that B cells were present in tumors, particularly plasma cells were present in tumors, in many tumors, and often when they produce IgG in red, associated with good prognosis. And there is a magnificent paper by the group of Ziv Schulman and Cell showing that when you label tumors with anti-IgG antibodies, of course, lymphoma are positive when they are B-cell lymphomas, but in solid tumors, you have many of them which are positive. The number one being renal cell cancer, we were lucky. Number two being liposarcoma. But in each tumor type, we have a labeling that is important for a subset of tumor cells. So last year, we proposed what we call the intratumoral immunity cycle, in which, in addition to an immune reaction that can be shaped in the lymph node, when in a tumor you have a tertiary lymphoid structure, you have a T cell zone where dendritic cells present antigen to T cells, CD4 and CD8, and also in B cell zone, where B cells can mature towards plasma cells. Plasma cells undergo selection amplification of their repertoire, affinity maturation, isotypic switching versus IgG and IgA, travel along fibroblastic conducts to the tumor cells, produce antibodies, IgG or IgA. When they are IgG, then macrophages or NK cells, maybe complement, can bind to these IgG, induce a certain level. It doesn't cure the tumor, but it uses a certain level of release of tumor cell death with reliefs of antigens, including intracellular antigens, which are targets for the T cells, and to which the T cells are not tolerant because usually they are not seen. And these immune complexes with cell membrane antigens or antigenic peptides are taken up in niches, the TLS themselves or immune niches, either by dendritic cells or by B cells, and presented to T cells. This is a formidable amplification signal that allows tumors with low mutational burden to increase their antigen presentation, and then when you come with anti-PD-1 or anti-CTLA-4, it diminishes the threshold by which is necessary to activate these tumors, and then increases the field of patients and tumor types that respond to immunotherapy. Also, these B cells, as T cells, travel in the periphery and take care of potential metastatic sites. So we are analyzing now what is the resistance of why not all TLS response positive tumors respond to immunotherapy, and this is ongoing work, but I chose not to conclude on that. I wanted to conclude on a positive note with lack of time. If you need a TLS and an ongoing immune reaction, then it is stupid to treat patients when they don't have a tumor anymore, because you don't have that. The surgeon has taken out the tumor, and you treat something that doesn't exist anymore. And now what appears is the neoadjuvant immunotherapy. People begin to treat patients with immunotherapy before the surgeon takes out the tumor, when all the immune reaction is in place. And you may have seen in this famous scientific journal called the New York Times, patients that have been with rectal cancer that have been treated with this approach, 100% have seen the tumor gone. Here it is in colorectal cancer, and as you can see here, now it has been extended. There has been 100% pathological responses, 60% complete pathological responses. So there's no tumor anymore seen by the pathologist. Here's an analysis from the Dutch group Christophe Blanc in metastatic melanoma patients. This is adjuvant after the tumor has been released, taken out, that are treated by immunotherapies. 57% are alive at 1 year. This is before 83% of the patients, no tumor. And now in the Netherlands, they don't use the surgeon to take out the tumor when clearly they see that the tumor has disappeared. They follow the patient. And this has really completely, and is going to change the fate of cancer. So my conclusion is that after a long period of doubt of the cancer scientists and clinical practitioner communities, the impact of the immune responses to cancer cells has been recognized as being a major partner of cancer control immunotherapies, have become a pillar of anti-cancer therapies, changing the life of many patients in almost all types of cancer. Modern immunotherapy is still in its infancy, and the use of novel monoclonal antibodies, novel immune checkpoint inhibitors, novel cells to be targeted, adaptive immune cell therapies, vaccines, will open new avenues to treat cancer patients at different stages of the disease, strongly enriching and modifying the clinical care of cancer patients. And remember, 25 years ago, nobody believed that it could even exist. And I would like to conclude to thank the people that I have cited the work. Of course, my wife, Catherine Sotes Fridman, with whom we do all this work together for, I don't want to say how many years. And the different participants, Maxime Mélan, Guilhem Pupier, Florent Petitpré, Étienne Becht, Théo Bouloudanian, Calvez, some are missing. Isaias Hernandez is not even here, and others, and of course a strong collaboration with clinicians and pathologists in France and in other countries. I thank you for your attention.